Van Helmont claimed he had uncovered the secret of life itself, and he even shared the formula. According to him, if you placed a dirty sweat-soaked shirt together with a handful of wheat grains inside an open container, then waited exactly 21 days, something extraordinary would happen. The sweat would somehow transform the wheat, and before long, fully grown mice would appear inside the jar, running, jumping, and very much alive. Yes, really. One of the most celebrated chemists of his era genuinely believed that a sweaty shirt and a pile of wheat could turn into living mice. The idea became known as spontaneous generation, the belief that life could emerge directly from non-living matter. For centuries, people accepted similar ideas. They believed maggots simply formed inside rotting meat, frogs emerged from mud, and living things could appear whenever the conditions were right. Today, it's easy to laugh at Van Helmont's experiment. We know those mice weren't magically created inside the jar. They were simply attracted by the grain. But beneath that strange conclusion was one of the greatest questions humanity has ever asked. How did lifeless, unconscious matter come together to become something that could think, feel, breathe, and eventually become us? If dirty laundry wasn't the answer, then what was the real recipe? What ignited that very first spark? How did life emerge from the young Earth's primordial chemistry? Welcome back to Uluse, everyone. I'm Hassan. Today we're setting out on another incredible journey as we tackle what may be the greatest mystery in the universe. Together, we'll search for the long-lost recipe that first gave rise to life billions of years ago. How did life actually begin? About 4.5 billion years ago, Earth was still an infant. Our planet had only just formed, and it looked nothing like the peaceful blue world we call home today. This wasn't a place where you could roll out a welcome mat and settle in. Scientists call this period the Hadean Eon, named after Hades, the ancient Greek underworld. And the name fits. Imagine opening your eyes on this newborn planet. The ground beneath your feet has barely cooled, covered by a thin, fragile crust that cracks with every step. Volcanoes erupt constantly across the horizon. Massive asteroids and comets rain down from space without warning. And the sky? Forget the clear blue sky we know today. Above you hangs a thick, suffocating haze of methane, tinting the atmosphere with eerie shades of. Or now, let's rewind the story even further, to a time before the first cell existed, before even the simplest organic molecules had appeared. We're traveling back to a barren, lifeless Earth, where the ingredients for life had yet to come together. To understand how scientists approached this mystery, we first have to visit an era before the scientific method existed, when people relied on observation alone to explain the natural world. Imagine living in ancient Greece and asking Aristotle a simple question: where do living things come from? His answer would have been immediate and confident. They arise spontaneously. This idea, known as spontaneous generation, dominated scientific thinking for nearly 2,000 years. As strange as it sounds today, it seemed perfectly reasonable at the time. People would leave a piece of meat outside. A few days later, it would be crawling with maggots. After the Nile River flooded, frogs and snakes seemed to appear from the mud left behind as the water receded. To ancient observers, the conclusion was obvious. Life was simply emerging from non-living matter. The belief was that some mysterious vital force could awaken lifeless material and transform it into living organisms. As we mentioned earlier, even in the 17th century, the Belgian physician Jean-Baptiste Van Helmont published what he believed was an actual recipe for making mice. According to him, all you needed was a dirty shirt, some wheat, and 21 days of waiting. For centuries, ideas like these weren't considered myths. They were accepted as scientific truth. Then the skeptics arrived. In 1668, an Italian physician named Francesco Redi suspected something wasn't adding up. He designed an experiment that was brilliantly simple. He placed meat into several jars. Some were left completely open. Others were covered with a fine gauze that allowed air to pass through but kept flies out. The results were impossible to ignore. Maggots appeared only in the open jars, where flies could land and lay their eggs. The covered meat still rotted, but no maggots ever formed. The maggots weren't coming from the meat. They were coming from the flies. It was a major blow to spontaneous generation, but it wasn't enough to end the debate. The decisive experiment came nearly 2 centuries later. In 1859, Louis Pasteur designed what would become one of the most famous experiments in the history of science, the swan neck flask experiment. Pasteur first boiled nutrient broth to kill every microorganism inside, sterilizing the liquid completely. Then he placed the broth into specially designed glass flasks with long curved necks, shaped like the neck of a swan. Air could still enter the flask, but dust particles and microorganisms became trapped in the bends of the glass before they could reach the broth. The result was astonishing. For years, the broth remained perfectly clear. Nothing grew. No life appeared. But the moment Pasteur broke off the curved neck, allowing dust from the air to fall directly into the liquid, microorganisms rapidly multiplied. Pasteur had demonstrated something profound. Life does not simply appear out of nowhere. Every living organism comes from another living organism. This principle became known as biogenesis. Pasteur's discovery transformed medicine and completely changed our understanding of infectious disease. But it also created an enormous scientific and philosophical problem. If every living thing comes from another living thing, then where did the very first living thing come from? The chain couldn't stretch backward forever. Somewhere, there had to be a beginning. It was the ultimate version of the classic chicken and egg question, and answering it meant looking beyond biology itself, down to the chemistry of matter. By the 1920s, 2 scientists working independently on opposite sides of Europe arrived at remarkably similar ideas. Russian biochemist Alexander Oparin and British evolutionary biologist J.B.S. Haldane proposed a bold new hypothesis that preserved biogenesis, while still explaining how life could have begun naturally. Maybe, they argued, the first living system didn't come from another organism at all. Maybe it emerged gradually from the chaotic chemistry of the young Earth. Simple molecules, given enough time and the right conditions, could slowly assemble into increasingly complex structures until something that behaved like life eventually appeared. Today, this idea is known as abiogenesis. Haldane famously described Earth's early oceans as a hot, dilute soup, a vast chemical laboratory where ultraviolet radiation from the sun, volcanic heat, and constant lightning supplied enormous amounts of energy. That energy could drive simple inorganic compounds to combine into more complex organic molecules. It was an elegant idea, but it was still only an idea. It needed evidence. That's when an ambitious young American graduate student stepped into the story. His name was Stanley Miller. In 1951, at just 21 years old, Miller attended a lecture at the University of Chicago, given by his Nobel Prize-winning advisor, Harold Urey. Urey was describing the atmosphere of the early Earth as a reducing environment, rich in hydrogen and other gases, with virtually no oxygen, perhaps resembling the atmospheres of planets like Jupiter and Saturn. As Miller listened, a single thought struck him like lightning. Miller couldn't stop thinking about it. Why don't we recreate those conditions in the lab and see what actually happens? He immediately went to Harold Urey with the idea. At first, Urey wasn't convinced. "It's a risky project, Stanley," he warned. "There's a good chance you'll get nothing at all. Don't gamble your career on it. You'd be better off working on meteorites or some safer research topic." But Miller refused to let the idea go. "Give me 6 months," he said. "If nothing comes of it, I'll move on." Urey finally agreed. In 1952, Miller assembled what would become one of the most famous experimental setups in scientific history. The apparatus was surprisingly simple. 2 glass flasks connected by a series of tubes. The lower flask represented the ancient ocean. Miller filled it with water and heated it until it produced steam. The upper flask represented Earth's early atmosphere. Into it, he introduced gases that scientists at the time believed dominated the young planet: methane, ammonia, hydrogen, and water vapor. Then came the crucial ingredient: electric sparks. Again and again, he discharged electricity through the gas mixture to simulate lightning flashing across the early Earth. What happened next surprised everyone. After the first day, the water had turned faintly pink. Within a week, it had become dark red and cloudy, almost like diluted blood. When Miller analyzed the liquid, he could hardly believe what he was seeing. It wasn't just a mixture of random chemicals. The solution contained amino acids, the fundamental building blocks of protein: glycine, alanine, aspartic acid. The essential ingredients of life had formed from nothing more than simple gases, water, and electrical energy. No living cells, no enzymes, no biology, just chemistry. The experiment became known as the Miller-Urey experiment, and its impact on science was enormous. Its message was both simple and revolutionary. Perhaps you didn't need anything supernatural to begin life. Maybe all you needed was the right chemistry and a source of energy. But science is a process of constant revision, and no idea remains unquestioned forever. As the decades passed, geologists and atmospheric scientists began re-examining what Earth's early atmosphere was actually like. By the 1970s and 1980s, growing evidence suggested that Miller's gas mixture might not have accurately represented the young planet. Instead of being rich in hydrogen and methane, early volcanic gases may have consisted mostly of carbon dioxide and nitrogen. That created a serious problem. When researchers repeated Miller's experiment using those revised atmospheric conditions, the results were far less impressive. Only tiny amounts of amino acids formed, and many of them were quickly destroyed by other chemical reactions. As a result, confidence in the classic primordial soup model began to fade. Some researchers even joked that the soup had gone cold. New ideas started to take center stage, but the story didn't end there. Stanley Miller defended his work for the rest of his life. After he passed away in 2007, his former student Jeffrey Bada discovered dozens of carefully labeled sample tubes from Miller's laboratory. Many had been sitting untouched since the 1950s and had never been analyzed using modern instruments. When scientists finally examined them with today's far more sensitive technology, they uncovered an astonishing surprise. Miller had originally reported producing only 5 amino acids. In reality, his experiments had generated more than 20. That completely changed how researchers viewed his work, and the surprises kept coming. Studies published during the 2020s suggested that even if Earth's atmosphere wasn't uniformly rich in methane and hydrogen, localized environments near massive volcanic eruptions could have created exactly the kind of temporary, lightning-filled, hydrogen-rich conditions Miller had envisioned. Then, in 2021, another study proposed something even more unexpected. The borosilicate glass used in Miller's apparatus may not have been chemically neutral at all. The glass itself could have acted as a catalyst, accelerating some of the reactions that produce these organic compounds. In other words, the primordial soup may still be on the menu. Perhaps it didn't fill the entire ocean. Maybe it existed only in isolated volcanic ponds, small lakes, or temporary pools where the chemistry was just right. Now, let's assume those amino acids really did form. Great. We have the building blocks, but a pile of bricks isn't a house. Likewise, a collection of amino acids isn't a living organism. Something still had to organize those molecules into a system capable of storing information, reproducing itself, and evolving. And that's where biology runs into one of its greatest paradoxes, the molecular version of the classic chicken and egg problem. In every living cell today, DNA serves as the long-term storage device for genetic information. Think of it as the instruction manual. Proteins, especially enzymes, are the machines that perform almost every task inside the cell. They copy DNA, build cellular structures, generate energy, and keep everything running. You can't build proteins without DNA because the instructions for making proteins are encoded in DNA. But you also can't copy, read, or maintain DNA without proteins because specialized enzymes carry out those jobs. So, which came first? The information, or the machinery that reads the information? For years, that question seemed impossible to answer. Then, beginning in the 1960s, scientists including Carl Woese, Francis Crick, and Leslie Orgel proposed a remarkable possibility. 2 decades later, groundbreaking discoveries by Thomas Seck and Sidney Altman provided powerful evidence that the idea might actually be true. Maybe the earliest life had neither DNA nor proteins. Maybe there was once a single molecule capable of doing both jobs. RNA. This concept became known as the RNA world hypothesis. Structurally, RNA is closely related to DNA. RNA is remarkably versatile. Like DNA, it can store genetic information. But unlike DNA, which exists as a stable double helix, RNA is usually a single strand. That flexibility allows it to fold into intricate three-dimensional shapes, behaving much like a protein enzyme. These catalytic RNA molecules are called ribozymes. In other words, RNA can play both roles. It can store information, and it can carry out chemical reactions. The RNA world hypothesis proposes a sequence that looks something like this. First, simple nucleotides formed within the primordial environment. Next, these nucleotides accumulated on the surfaces of certain clay minerals, where they lined up and chemically linked together into longer chains. Among billions upon billions of random RNA strands, one eventually adopted a structure capable of making imperfect copies of itself. Once self-replication existed, Darwinian evolution could begin. Some RNA molecules copied themselves more efficiently than others. Random mutations created variation, and natural selection favored the fastest and most accurate replicators. Over immense stretches of time, this primitive RNA world grew increasingly sophisticated. Eventually, DNA emerged as a more stable way to preserve genetic information, while proteins took over as highly specialized molecular machines capable of performing chemistry far more efficiently. RNA never disappeared. Instead, it settled into a supporting role, one it still performs inside every living cell today. One of the strongest pieces of evidence for this idea is hiding inside your own body. Every protein your cells produce is assembled by ribosomes, the molecular factories responsible for translating genetic information into proteins. And at the very heart of every ribosome, the most critical catalytic component isn't a protein at all. It's RNA. That ancient RNA core is often described as a molecular fossil, a surviving relic from a time when RNA may have ruled biology on its own. But like every major scientific hypothesis, the RNA world isn't without its challenges. RNA is an extremely fragile molecule. Ultraviolet radiation easily damages it. Water tends to break RNA strands apart through hydrolysis. Even under favorable conditions, maintaining long RNA molecules without them falling apart is chemically difficult. Making RNA is another obstacle. Its building blocks, the nucleotides, are considerably harder to produce under natural prebiotic conditions than amino acids. Molecules such as cytosine and uracil are especially challenging to synthesize without the help of living organisms. In 2009, chemist John Sutherland and his research team demonstrated a promising pathway in which nucleotides could form through a network of interconnected chemical reactions, rather than through simple step-by-step synthesis. That work solved part of the puzzle, but it didn't close the case. Scientists still don't know exactly how an RNA world, if it truly existed, got started. Perhaps RNA wasn't the first genetic molecule. Simpler relatives such as TNA or PNA may have come before it. Or perhaps RNA never existed in isolation at all. Maybe information storage and metabolism evolved together from the very beginning. At this point, no one can say for certain, and that uncertainty brings us to another fundamental question. Even if RNA could store information and replicate itself, where did it get the energy to do any of it? Copying molecules isn't free. Every chemical reaction requires an energy source. Lightning is unpredictable. Sunlight can be destructive. Life needed something continuous, reliable, and available for millions of years. To find that energy source, scientists looked to one of the most extreme environments on Earth, the deepest parts of the ocean. In 1977, researchers exploring the seafloor discovered towering hydrothermal vents now known as black smokers. Picture descending nearly 2.5 miles beneath the ocean's surface into complete darkness. The only light comes from your submersible. As the beams cut through the black water, enormous chimney-like towers suddenly emerge from the seafloor. Some rise as high as an 8-story building. They're pitch black, and from their peaks billow dense clouds that resemble smoke pouring from giant industrial smokestacks. It's superheated water, reaching temperatures of around 750 degrees Fahrenheit, packed with dissolved minerals that instantly cool as they collide with the near-freezing seawater. The result is a dramatic black plume that gives these vents their name. At first, scientists thought these hydrothermal systems might provide exactly the steady energy source needed for life's origin. But there was a problem. Black smokers are incredibly hot. They're also highly acidic. For delicate organic molecules, those conditions may be more destructive than helpful. Then, in 2000, researchers exploring the mid-Atlantic ridge discovered something completely different. A remarkable hydrothermal field now known as the Lost City. Instead of dark, scorching chimneys, these vents were pale in color, cooler, and strongly alkaline. That discovery inspired biochemist Nick Lane and geologist Mike Russell to develop one of the most influential origin of life hypotheses proposed in recent decades. Their central idea was surprisingly simple. Life may have begun because of a difference in proton concentration. Inside these alkaline vents flowed water that contained relatively few protons. Outside, the surrounding ocean was much more acidic. During the Hadean Eon, Earth's oceans likely contained abundant dissolved carbon dioxide, making them significantly richer in protons than they are today. These two very different fluids met inside the microscopic, sponge-like mineral walls of the hydrothermal vents. On one side, a high concentration of protons. On the other, very few. That difference created a natural electrochemical gradient, a voltage, in the simplest sense, much like the positive and negative terminals of a battery. Nick Lane argues that this is the crucial piece of the puzzle. According to his hypothesis, the microscopic pores inside these alkaline hydrothermal vents acted as nature's first incubators. Within those tiny mineral chambers, the steady flow of protons supplied exactly the kind of continuous energy needed to drive simple molecules into forming more complex organic compounds. Much like water flowing through a dam spins a turbine to generate electricity. What's truly remarkable is that this isn't just ancient history. Your own cells still work the same way. Inside nearly every cell in your body are mitochondria, the tiny structures responsible for producing the energy that keeps you alive. They create ATP by pumping protons across a membrane and then allowing them to flow back through molecular machinery. The basic principle is virtually identical. In a sense, we're talking descendants of ancient hydrothermal vents. Pretty amazing, isn't it? For years, this idea remained both fascinating and frustrating. It was elegant on paper, but proving it experimentally seemed incredibly difficult. Then, in 2024, a research team led by Thiago Alter Ferrara in Brazil published a study that brought the hypothesis one step closer to reality. The researchers built a laboratory reactor designed to mimic conditions at the floor of the Hadean Ocean. On one side, they placed acidic seawater. On the other, alkaline vent fluid. Between them, they inserted iron-sulfur minerals, the same kinds of minerals found inside hydrothermal vent structures. There were no living cells, no enzymes, no biology whatsoever, just rocks, water, dissolved gases, and a natural chemical gradient. The results were extraordinary. Using nothing more than the difference in pH and the catalytic properties of those minerals, the system converted carbon dioxide into formic acid and acetic acid. That may sound like ordinary chemistry. It isn't. Acetic acid, the same molecule that gives vinegar its characteristic acidity, is one of the most fundamental compounds in metabolism. Deep inside every one of your cells, ancient biochemical pathways still rely on closely related chemistry to generate energy. Ferrara's experiment suggested something profound. Long before life existed, nature may already have been rehearsing the chemistry of life. The rocks were doing chemistry. The water was doing chemistry. The stage was being prepared before the first living actor ever appeared. If this interpretation is correct, metabolism didn't emerge after life. Metabolism may have paved the way for life itself. It's one of the strongest pieces of evidence supporting what's known as the metabolism first hypothesis. But while we've been looking downward into oceans, volcanic vents, and ancient chemistry, there's another possibility we shouldn't ignore. What if some of life's ingredients didn't originate on Earth at all? What if they came from space? This idea is known as panspermia, a term that literally means seeds everywhere. The hypothesis doesn't necessarily claim that intelligent aliens planted life on Earth. Instead, it suggests that the ingredients for life, or perhaps even primitive microorganisms themselves, may have traveled across the cosmos aboard asteroids, comets, or microscopic dust particles before eventually reaching young planets. It sounds like science fiction, but the evidence behind it is surprisingly tangible. One of the most important discoveries happened on September 28, 1969. On that day, a massive meteorite exploded over the town of Murchison in Victoria, Australia, scattering thousands of fragments across the surrounding countryside. Scientists quickly recovered the pieces and began analyzing them. What they found was astonishing. The meteorite belonged to a rare class known as a carbonaceous chondrite, a type of space rock rich in organic material. Inside it, researchers identified more than 90 different amino acids. Life on Earth uses only about 20. They also discovered sugars and nucleobases, the molecular letters that make up DNA and RNA. Finding those compounds inside a meteorite was extraordinary enough. Then came an even bigger surprise. The material was older than our solar system itself, preserving chemistry that dates back roughly 7 billion years. That meant complex organic chemistry wasn't unique to Earth. It had been taking place across interstellar space long before our planet even existed. One of the most curious details about the Murchison meteorite wasn't what scientists saw. It was what they smelled. Researchers reported that freshly fractured pieces released an unusual odor. Some described it as wet hay or tar. Others compared it to Brussels sprouts, while a few said it resembled rancid rancid peanut butter. Those strange smells came from volatile organic compounds that had remained trapped inside the meteorite for billions of years before finally escaping into Earth's atmosphere. Space, it turns out, may not be as sterile as we once imagined. It's filled with complex organic chemistry. Another fascinating version of panspermia is called lithopanspermia. The idea goes like this. Mars is smaller than Earth, meaning it cooled much more quickly after it formed. If liquid water appeared there earlier, then perhaps life also began there first. Later, a massive asteroid impact could have blasted Martian rocks into space. Some of those rocks, carrying dormant microorganisms sheltered deep inside, might eventually have landed on Earth. If that sounds unbelievable, remember this. We already know that rocks can travel naturally from Mars to Earth. Scientists have identified dozens of Martian meteorites in collections around the world. Whether any ever carried living organisms is still completely unknown. That's one reason why missions from NASA, China, and other space agencies continue searching Mars for evidence of ancient microbial life. But it's important to keep one thing clear. Even if panspermia turned out to be true, it wouldn't actually solve the biggest mystery. It would simply move the question somewhere else. If life arrived on Earth from another world, then how did life begin on that world in the first place? Panspermia doesn't eliminate the mystery. It simply moves it somewhere else. If life arrived on Earth from space, then the obvious question becomes, how did life begin out there? Eventually, every explanation leads us back to the same place, to chemistry and to abiogenesis. So after exploring primordial oceans, hydrothermal vents, and meteorites from deep space, let's tackle the question that's probably been sitting in the back of your mind this entire time. Maybe you're thinking, this all sounds fascinating, but isn't it still impossible? Can atoms and molecules really organize themselves into something as complex as a human being, purely by chance? It's an understandable question. Probability has always played a huge role in this debate. One of the most famous critics of abiogenesis was the British astronomer Fred Hoyle. He disliked the idea that life could emerge through natural processes, so much that he came up with a memorable analogy. He argued that the odds of life appearing by chance were about the same as a tornado sweeping through a junkyard and leaving behind a fully assembled Boeing 747. It's a striking image, a violent storm, a pile of scrap metal, and suddenly, a perfectly functioning airplane. You've probably heard another famous analogy as well, the infinite monkey theorem. Imagine an infinite number of monkeys sitting at typewriters, randomly hitting keys forever. Given unlimited time, could one of them eventually produce Shakespeare's Hamlet letter for letter? Mathematically, the answer is yes. But in practice, physicists point out that the universe isn't old enough for those monkeys to even finish writing the opening line. At first glance, these comparisons seem devastating, but they contain a fundamental misunderstanding. Chemistry is not a drunken monkey randomly smashing keys. Chemistry follows rules. Carbon atoms don't wake up one morning and decide to behave differently. Under specific conditions, certain atoms naturally bond with certain other atoms because the laws of physics leave them little choice. Think about Hoyle's junkyard analogy again. It assumes every piece is flying around completely at random. But real chemistry doesn't work like that. It's more like every bolt is naturally attracted to the correct nut, every connector fits a specific socket, and every component has preferred ways of assembling itself. The amino acids produced in the Miller-Urey experiment didn't appear because of blind luck. They appeared because the underlying chemistry favored their formation under those conditions. One of my favorite authors, Douglas Adams, captured this idea beautifully with a story from The Hitchhiker's Guide to the Galaxy. He imagined a puddle waking up one morning and looking around in amazement. "Wow," the puddle says. "Look at this hole. It's absolutely perfect for me. The curves fit me exactly. The depth is just right. Surely this hole must have been designed specifically so I could exist inside it." The hole wasn't shaped for the puddle. The puddle simply took the shape of the hole. Adams argued that we often make the same mistake about the universe. We look around and conclude that the universe seems perfectly designed for us, but perhaps the opposite is true. We evolved to fit the universe, not the other way around. From this perspective, the emergence of life becomes less like an impossible miracle and more like the natural outcome of a planet that possesses three simple ingredients: rock, liquid water, and carbon dioxide. Give those ingredients enough time and perhaps life isn't an extraordinary accident at all. Perhaps it's what chemistry naturally tends to do. For hundreds of millions of years, the young Earth functioned as an enormous electrochemical reactor, running countless chemical experiments simultaneously across its oceans, coastlines, volcanic islands, and hydrothermal systems. Given enough opportunities, maybe life wasn't incredibly unlikely. Maybe it was almost inevitable. And if that's true, then another question naturally follows. If life arises whenever the conditions are right, are we really alone in the universe? That brings us to one of the most famous equations in modern astronomy, the Drake equation. Don't worry, we're not going to spend time working through mathematical formulas. The story behind the equation is far more interesting than the equation itself. It's 1961. The location is Green Bank, West Virginia. Radio astronomer Frank Drake has invited a small group of scientists to what would become the first conference devoted to the scientific search for intelligent extraterrestrial life. Back then, publicly talking about searching for aliens wasn't exactly a great career move, so the meeting was intentionally kept low profile. The guest list was extraordinary. A young Carl Sagan, Nobel Prize-winning biochemist Melvin Calvin, and neuroscientist John Lilly, who had become famous for his ambitious attempts to communicate with dolphins. The group even gave themselves a name, the Order of the Dolphin. No, that's not a joke. That was the actual name they used. Lilly was so fascinated by dolphin intelligence that he believed learning to communicate with another intelligent species on Earth might prepare humanity for one day communicating with an intelligent species somewhere else in the cosmos. During the meeting, exciting news arrived. Melvin Calvin had just been awarded the Nobel Prize in Chemistry. Champagne bottles were opened. The celebration began, and amid that atmosphere of excitement, conversation, and possibility, Frank Drake walked to a blackboard and wrote down what would become one of the most famous equations in the history of science. The Drake equation asks a deceptively simple question. How many galaxies are there? How many stars do those galaxies contain? How many of those stars have planets? How many of those planets could support life? And perhaps the most important question of all, how many civilizations survive long enough to become technologically advanced and communicate with others? In other words, how many intelligent civilizations might exist in the universe? In 2016, astrophysicists Adam Frank and Woodruff Sullivan revisited Drake's famous idea from a different angle. Rather than trying to estimate how many civilizations exist right now, they asked a broader statistical question. How unlikely would technological life have to be for humanity to be the first and only civilization the universe has ever produced? Their conclusion was astonishing. Only if the odds of a technological civilization emerging were less than about 1 chance in 10 billion trillion would it become statistically reasonable to think we're truly alone. That's such an unimaginably tiny probability that it's like winning the lottery thousands of times in a row. If the laws of physics are the same everywhere in the universe, and every observation so far suggests that they are, then the fact that we exist strongly hints that life may have emerged many times across the cosmos. The most likely explanation isn't that we're alone, it's that we're separated by enormous distances, or perhaps we're simply not listening in the right way. Think about it. Humanity has been broadcasting radio signals into space for only a little over a century. That means our technological footprint has expanded just over 100 light years from Earth. Now, compare that to the size of our own galaxy. The Milky Way stretches roughly 100,000 light years across. Our radio bubble is still a tiny ripple in an unimaginably vast cosmic ocean. Today, we've traveled through one of science's greatest mysteries. Looking back, what we have isn't a complete answer. It's a puzzle whose pieces are finally beginning to fit together. The hellish Hadean Earth, with its orange skies, toxic atmosphere, emerald green oceans, and the smell of sulfur hanging in the air. The primordial soup, where lightning and volcanic energy may have driven the first complex chemistry. The possibility that RNA learned to copy itself. The microscopic chambers inside ancient hydrothermal vents, where natural proton gradients may have powered the earliest metabolism. Stanley Miller's Crimson experiment. Bio-2. Nick Lane's natural battery. Diego Ferrara's laboratory reactor, where lifeless minerals recreated pieces of ancient metabolism. The Murchison meteorite, carrying organic molecules that formed before our own solar system even existed. Every one of these discoveries points in the same direction. Life doesn't appear to violate the laws of physics. It appears to emerge from them. Perhaps the universe isn't simply filled with stars, planets, and empty space. Perhaps, whenever the conditions are right, the universe naturally organizes matter into chemistry, chemistry into biology, and biology into minds capable of wondering where they came from. The atoms inside you have traveled an extraordinary journey. The iron in your blood was forged in stars that exploded billions of years before Earth existed. The electrical energy flowing through your cells still depends on the same proton gradients that may have powered the earliest chemistry deep beneath the ancient oceans. Every cell in your body is the product of an unbroken chain of life, stretching back nearly 4 billion years. When you look in the mirror, don't just see another human being. See the latest chapter in one of the longest stories the universe has ever told. A story that began when a handful of simple molecules somehow learned to make imperfect copies of themselves. Where that story goes next may depend on us. If you enjoy the videos here on Holocene, consider supporting the channel so I can keep making more deep dives like this. You'll find the membership link in the description below. Until next time, stay curious, and don't forget that the same spark that first appeared nearly 4 billion years ago is still alive inside you. I'll see you in the next video.